Okay, I think we can get started, and those people that file in a little bit later can catch up. So we have, again, a relatively full agenda for today. I'm going to give just a brief introduction, and then we'll hear about some data from Jerome. He's got some information and some new data on some of the potency of our compounds and a couple of cell lines. Then we'll go through the usual Alex, Sue, and David SAR update, and then we'll come back and talk about future plans and how we're gonna move forward. Again, I think this month should be, we should have time at the end for significant discussion, but I always underestimate how long people take to present, including myself. So, so with that, let's get started. So again, I always like to show this slide when we start. There is this dire unmet need to try to treat DIPG, and unfortunately, we do have some bad news about Aubrey, who, if you all remember, we did the Lemon Face Challenge. Unfortunately, she did pass away on August 15th after a year-long battle with DIPG. So it just reminds us about how important our work is that we're doing, and how much we need something to treat these kids. So with that, let's just move forward. So action items from the previous meeting. I think this was actually from 2 meetings ago. This first one, Jerome, was to check with Stanford about accessing additional cell lines. And I don't know if you want to comment, Jerome, on that.
Yeah, I mean, I got a new cell line, DIPG 36, and you'll see I had the chance to test one of the M4K compounds in it, and it grows quite well. So okay. I got one more cell line.
Okay, great. So at the last meeting, we had talked with Javad Nazarian from the DIPG Consortium, and we were putting in place an MTA to be able to send our compounds there so that he can evaluate them in his cell lines and also in his microdialysis in vivo model, where you can get real-time information about brain exposure. So that has been taken care of. We've sent the compound, and we're working closely with Sri Devi, who is a staff scientist in Dr. Nazarian's lab, and we'll see when that data comes.
I have a question just for my narrative. How long did that MTA take to execute?
What? Oh, and maybe a month?
Yeah.
Yeah, it was about a month.
And was there any negotiating back and forth?
Not really. Not really. It was just
process.
Just getting into someone's
hands and waiting.
Okay. So, I don't think, my recollection is there wasn't a lot of pushback. There's some cleanup on some few things.
Okay.
Which is interesting because there are clauses around not patenting, right? So there was no pushback.
And that was accepted. Yeah.
So, but it had been explained well. So
yeah. Okay, so that's some exciting work that I look forward to seeing some data for in the not too distant future. As well, we talked about reaching out to the FOP network to identify a potential lab to test our compounds. I can tell you that we've been able to identify one lab at UPenn. Dr. Eileen Shore is actually very excited to test our molecules. We're working on getting an MTA put in place to be able to enable that. And once that's in place, we'll be able to send her some of our compound and be able to evaluate our compound in our FOP model. I'm hoping at one meeting in the not too distant future, either the next one or perhaps a little bit further out, that she'll actually present at one of these meetings and talk about.
Is there a way to send the compound on a handshake, over the telephone handshake, and then get the MTA executed? I realize it exposes us a teeny bit and stuff. But is that possible?
Well, I think at this point, I think Owen, you've already sent the MTA. Yeah. She has it, and so okay. I'm not sure that it's going to save that much time. We need to finish scaling up the compound before we will be able to send it anyway. So.
And she has to schedule her mice and her animals, etc. So I don't believe it's the rate limiting step this time. Okay. Should it become, then we'll revisit it.
Okay. Yeah. So hopefully we should be able to get some of that data in the not too distant future. We talked about scheduling a follow-up meeting with the Charles River ADME team to discuss the next steps. If you recall at the last meeting, We talked about some potential opportunities in 2009 and what the right path forward would be in terms of dose escalation, in terms of an MTB, and in terms of testing the compounds in this FLP model. And we did have that meeting. Sue was able to organize that, and we had that meeting. And I think we came up with a good plan, and I'll discuss that a little bit later in the presentation. CRL, Sue was going to get quotes. We talked about running both M4K 2009 and 3007 using the proposed Charles River PK process, looking at both plasma and brain. And so we have quotes for that. We're going to move forward with that and see how that data compares with the data that we've generated at our other vendor where we previously generated the data. So it'll be really interesting to see especially 3007, because this is another interesting compound that I think has the potential to move forward. So that's some great data, and hopefully we'll be able to get that in the not too distant future as well. I was looking at getting quotes for scaling up 10 grams of M4K 2009. We did get quotes. I can tell you that the price was pretty expensive, mostly because there was a significant cost for the starting materials. The being able to selectively Suzuki in part of the molecule and then put the other half in. You need an asymmetric pyridine, which is relatively costly. So we decided not to go that route and to try to explore other chemistry using cheaper starting materials internally to be able to make that compound. And I think Sue and the Charles River team used the dibromo compound, the symmetric compound, and that's some chemistry that we're hoping to look into. And we're still trying to get the MDCK and DR1 assay online. Ahmed, I don't know if you want to comment. We're still waiting.
Yeah, we're still waiting. We had a hiccup because the plate were not available for some time, both from the manufacturer. We had to wait. We got them now, so it is on their way right now.
Great. And yeah, I mean, I think we're close as well to finalizing this ALK2 database. Internally, I have a meeting scheduled for Wednesday with our internal team, and we can hopefully get that rolled out in before the next meeting, and certainly that will help a lot when we start putting together the reporting. And with that, the reporting for our OICR grant, the CTIP grant, is due on October 5th. I just got the paperwork just the other day, so thanks, Al.
Yeah. Thanks in quotation marks.
Yeah, but so we need to put together a progress update for the project. It's a maximum of 8 pages, and I'll let people read this on their own. I think everyone should be able to access the slides, but it's a fairly in-depth report, and I will be reaching out to various people for contributions, and I think if we split the work up, it should be less onerous on all of us. So with that, I think that we can, I don't need to say too much more about the reporting, but it is an important element of the project since we are getting a significant amount of funding from OICR. I think, this is the least that we can do. We will also be presenting the project in November to the TPAC Advisory Committee, which looks after these projects that are funded by OICR. So that will happen in November as well. So again, I'll be reaching out for information and data to be able to support that presentation. Now, we do have a couple of newer people joining the project, and so I did want to go back and just kind of review the project flow scheme and the target product profile that we're going after. So we're going after a brain-penetrant ALK2 inhibitor for the treatment of DIPG. We have several different collaborators. We've got Charles River and OICR who are making analogs at this point. We're testing the compounds against ALK 1, 2, and 5 at RBC, and RBC Reaction Biology has kindly donated their assays to be able to support the project. We screen from time to time and just spot check molecules at RBC against the relevant DIPG mutants that they have. And so far, if we're active against wild type, generally we're active against all of these mutants. And so primarily we're screening just wild type ALK2 and just spot checking key compounds here. SGC Oxford is looking at getting X-ray structures for our compounds against ALK2 and ALK5, and they're also testing our compounds in cell-based assays. They have a nanobret ALK2 assay and a luciferase-based ALK5 assay at this point. As compounds progress through the cascade, if we're kind of less than 10 nanomolar in the biochemical assay and less than 100 nanomolar in the cell potency assays, then we kind of move in and we start gathering some in vitro ADME data. Internally here at OICR, we're looking at liver microsome stability in the presence of mouse and human liver microsomes. We're also running KCO2, and eventually, as we mentioned, we'll get the MBCK and DR1 assay up and running. And then we're outsourcing the PK, and if the PK looks good, then it moves into looking at whether or not we actually get compound into the brain. And then RBC is also looking at 5 kinases for us on a monthly basis that can be these mutants or some of the known off-target kinases that we hit, like RIPK2 or TNIC. And this is something that we're just starting to capitalize a little bit more on. And then, we've got folks like Javad and Jerome who are looking at proliferation and the the effect on phosphosmad and the ID1. I think we'll see some interesting data there today. And if the compounds continue to look good, they also go into more advanced in vitro ADME, looking at protein binding, brain tissue binding, CYP, HERG, Ames, the kind of standard assays to be able to nominate a potential clinical candidate. And we've also, at least for one compound so far, put a compound through a more extensive kinome panel to be able to see what other off-targets we're hitting. Now, we haven't moved compounds further than this yet, but we're hoping to. And, I mentioned the Eileen Shore collaboration for the FOP model. And we've got several options that we're looking at for the DIPG models. I know, Jerome, you've got a model that you're trying to develop. Chris Jones and Angel have another, the DIPG 007 model that, I think is certainly a good option. And so we're looking at how best to evaluate compounds here, but things are moving through this pipeline, and we're starting to get options to be able to test the compounds. So I think that all of that is very encouraging. So with respect to the target product profile, obviously we want something that's potent and selective, that has good in vitro ADME profile without a HERG liability or an Ames liability, with good bioavailability, good exposure in the brain, and also, in these sorts of projects where you're targeting the CNS, the unbound fraction has traditionally been accepted to be that which is the most relevant concentration to look at. So we're looking at trying to be above a 3-fold window with respect to the cytotoxic concentration against the DIPG cell line that we're going to move in vivo, at least to look at an efficacy study. So that's something that we continue to need to look at and evaluate. And then in an efficacy in a mouse model, we want to look at reduction in tumor volume as well as the standard Kaplan-Meier, improve survival. Of course, we want a good therapeutic index with no toxicity. We want to be able to demonstrate that our compounds are acting as they should and impacting the downstream markers of ALK2 inhibition, which is the phosphosmad 91 expression. And then hopefully we can identify a potential peripheral biomarker. And so that's the target product profile that we're going after. And with that, I think we can move back to the agenda, and we'll get an update from Jerome. Okay? And I'll come back later and discuss some of the profile and next steps. So let me just open Jerome's, which is this one.
Okay, so you've seen some of, or maybe most of these data before, which is kind of like a little update and some new things as well. So we've been basically testing some of your compounds in patient-derived cell lines. All of those so far have been obtained from Stanford. So the 4 cell lines that have been tested so far are listed here. And there's one that does not have an ACVR1 mutation. That's kind of like a useful control, I think. And 3 that are ACVR1 mutants, including a new one, the DIPG 36. They all have a glycine 328 mutation, but interestingly, it's a different substitution in all 3 cell lines, which is, I guess, nice because it allows us to compare the effect of this mutation. But it would be nice also to look at other mutated residues if the cell lines are available. I have confirmed the presence of the mutation at both the genomic DNA and the RNA cDNA level by sequencing in DIPG4 and DIPG21. I haven't done that yet for DIPG 36. So this is these are the profile of the response to kind of like some of the more interesting compounds. And you see that in general, they seem to inhibit the growth or the viability of ACVR1 mutant cells. I just tested the DIPG 36, the newest cell line with the 2009 compound, which is the most promising from what I understand. And there seems also to be kind of like a similar effect, but there's probably a little bit higher IC50 in that cell line. Although, again, this is one experiment, so should be repeated.
What's the time course?
This is so the cells are plated on day 1. On day 2, the drug is added, and 4 days later, this is added. Yeah. And the profile, at least in for most of the compound, I would say it would be consistent with the cytostatic effect. And this is something I can come back to when I show a little more data.
Are you seeing the efficacy track with potency?
I haven't done that comparison directly. Yeah. That's interesting. I should get that together and see if there's a correlation.
It doesn't correlate perfectly, no. I mean, essentially all of these are of pretty similar potency. So it's, I think given that it's a cell-based assay.
And I have another question. We've been chatting a lot about the conditions in which the cells are grown. Are these grown in like 5 times glucose normal and in 20% oxygen?
Yeah, so this is 20% oxygen, but in terms of glucose, so I should go back and see, this is cultured in defined medium. It's called the tumor stem medium, which contains like a bunch of growth factors. This is the conditions that were optimized at Stanford. Like there's a PDGFA.
Optimized for growth.
Yes.
Okay.
Yes, optimized for growth, and so this is based on these experiments, the calculated approximate IC50s for the different compounds. So, 2009, it varies a bit between cell lines. I put the DIPG 36, it couldn't really calculate accurately because as you see, the curve doesn't plateau at 10 micromolar. So, yeah. And then,
So you're not factoring, how are you factoring in the fact that some of these are not completely?
Yeah, unfortunately.
It's not reflected in these numbers.
Yeah, it's not reflected. So this is like the kind of automated calculation that GraphPad does when you input the data, right? So of course, if your curve doesn't plateau down, then it has a hard time calculating the IC50.
I think we should flag it somehow.
In terms of,
because like 043 looks great. You're good, you go back.
Yeah.
It's the curves that I think are the most.
You see?
I see.
I see what you mean, So this is, This would be for, exactly, this would be probably like a cytostatic, pure like cytostatic effect. Like, so, yeah, whereas in some cases, for example, in the case of this compound, you probably, if you look at DIPG 21, for example, you probably have some cytotoxicity as well, because of the rate at which these cells grow. You don't expect to reach like such a low value in this assay, you know, if it has only cytostatic effect, where it should be more like kind of around something like this. I mean, of course, this is estimated, so there's some variability there, but like this is probably cytostatic, this one as well, and this one as well.
I spent my whole other half of my life telling people that we need to do negative controls, we need to have SAR. So we have a bunch of active compounds that kill cells. You know, ain't surprising, is it?
Yeah.
Not a kinase inhibitor that kills cells.
No, I think, but I think it's important to show this information, and especially the information that he's about to show. I think that's the more relevant information. I mean, we do need to show that we will kill or at least stop growth of these cells.
Yeah, but
no, it doesn't kill everything, right?
No,
the other blue line, the blue cell,
it's not a perfect negative control, but it was intended to be, because the blue line, this one does not have an ACVR1 mutation. So of course these cell lines, they differ in many respects, not just the presence of the ACVR1 mutation. So it's not a great negative control, but at least the fact that you see differential response, I think.
But we must have a molecule that's inactive. You guys are good at making those.
Yeah, we do. I haven't tested that, but
we've supplied it. Yeah. We've given it to them.
Okay.
I appreciate the vote of confidence.
It's a Mark Bunny's line. It's not mine. I can't take credit for it.
I can definitely test this in the database. I just have to retweet it.
Yeah. So it's a structurally similar analog that is inactive. So it'll be good to show. But the fact that most of these compounds show a similar profile across all the cell lines, I think is good. I mean, we know for M4K 2009 that it's quite selective. We've got the full kinome there. For the other compounds here, we don't. So something like 2045 that's more active against the DIPG 21, that might be maybe that's not as quite as selective. It's hitting another kinase that could have an effect. So, I mean, it's an initial data set, and I think something that is good to have. But I think your gene expression data, the expression data.
So if we go to that, we did some experiments to look at target engagement. So phosphosmad and phosphoric was included here because this experiment also looked at this compound 6201, which is an inhibitor of ACVR1, but also of MEK1. And as you can see, basically compare also with the positive control LDN. These tested M4K compound, they all inhibit phosphosmad. The concentration that were picked here were approximately the IC70, but calculated based on the dose response curve.
Yeah. And
24 hours.
Eventually, for 2009, it might be nice to show a dose response.
Yeah, I try to see there. Yeah, I mean, of course, because we don't know.
Yeah.
Killing is nicer, but to me that's more relevant that you're engaging the target and the stuff.
Exactly.
If you go back on the previous slide, maybe the true out to phenotype is that shallow curve.
Yeah.
Not because they're killing for some of those deeper compounds, it might be doing other things. So.
Yeah. I have data specific with killing as well. Like, so this is an assay looking specifically at cell death. We knew that E6201, that the dual MEK1 and ACVR1 inhibitor kills cells. So this is confirmed in these assays. If you focus on this one, because this one has been repeated more times in the DIPG4 cell lines, but you can see that none of the M4K compounds induce detectable apoptosis. Another interesting thing, I don't show it on the slide, but the cells themselves, they actually change their morphology when they're treated with the M4K compound, and it all looks the same regardless of which compound is used. It's just the shape of the cells. I don't know what it means, but it seems they all have a similar effect on the cell, and that's not related to apoptosis. And this is something that Javad was mentioning as well, like a couple of meetings ago, about being careful or suspicious of compounds that just kill cells, right? So I think these data suggest that that's not the main thing that these compounds do. And so to look further at target engagement, then I measure the expression of downstream downstream target genes, so ID1, ID2, and ID3. The regulation of ID2 seems to be more complex, and that's dependent on ACVR1, and we've seen that in several different contexts and experiments. But if you look at ID1 and ID3, you can clearly see suppression of the gene. This is in the DIPG4 cells. Similarly, DIPG21, but in this case, ID2 goes down, interestingly. I don't know why, but again, these cell lines they differ in many respects, not just in the ACVR1 mutation. And the DIPG6, so this is the one that's our ACVR1 wild type, and you see a much more modest effect. I mean, it's possible that, there's some BMP signaling going on in these cells, right? But it's not driven by ACVR1 mutation. So yeah, that's what I have so far. And so just one comment about the DIPG36. When I met Michelle in Denver, she said that she thought that this cell line was xenographed. So I tried to confirm that with their lab after, and they weren't sure. So I have to test it myself, and it's going to take a little while, but I hear them to do that right now.
Okay, great.
All right, this is Paul. I have a question. If apoptosis is not going to kill the cells, and you think that a lot of the cytotox is from general cytotox, what's the kind of anti-cancer phenotype we're going to need to see?
Well, that's a good question. I mean, I guess our mission right now is to develop the best compound we have or we can to test that hypothesis, and that's been the whole basis of the project up to this point. Again, these are cell lines, as Al mentioned, they've been selected to grow well, and so but the hypothesis is that giving an OP2 inhibitor should treat the disease. So we're not seeing a lot of cell killing at this point. Maybe some cytostatic effects.
Yeah, there are many other aspects to that as well. Like you could have effects on the tumor cells that's not reflected by just cell killing, but could still be anti-tumorigenic, like causing them to differentiate or something like that.
So
knocking down the target has been shown in the literature.
So is it ultimately going to be an experiment to
right?
show that we think this mechanism actually kills DIPG?
Yeah, well, it's been shown in the literature by Chris Jones and some other folks at ICR and other institutes that if you knock down some of the DIPG cell lines with shRNA, that you do get some cell kill. I haven't seen whether or not that impacts in nexin staining or what that mechanism of cell kill is, and I'm not sure that has been disclosed or even. But based on the literature data that's out there, we and based on the project that was submitted, our goal is to identify the best compound as quick as possible to be able to prove or disprove the hypothesis that DIPG is going to be sensitive to an OP2 inhibitor.
I guess the question is that going to be in animals or in people, or when is that really going to be answered?
Well, again, I think hopefully we're going to be able to show it in an animal model because that's the only way we'll ever really get into humans. We're also looking at moving the compound into an FOP model, which is well demonstrated in the literature to be susceptible to an OP2 inhibitor. And so, if our compounds are active against that molecule or against that disease, and we're impacting the downstream biomarkers like the SMAD, like the ID1, and so we can demonstrate that we're on target, that we have an effect in vivo on a disease that we know is reliant on this kinase, then we'll be comfortable moving that molecule into a DIPG model, which again, I think even the DIPG models at this point are less well validated. And so, you even see across these cell lines, we're seeing some differences. So, you know, it's a challenging project so far.
Yeah, I mean, I think the phosphorus SMAD looks really good. A really robust effect. It just, yeah. I wonder if we can convince anybody else to just put it in patients if it's safe and it knocks that down if that's enough.
Yeah, I don't know. I mean, I guess that's something, you know, hopefully we'll be able to show in true in vivo efficacy in a model. I mean, the model that's been used so far for ALK inhibitors was a DIPG 007 model, and we don't have this data yet, although I think, Alex, you generated some DIPG 007 data, right? That shows that we're also impacting SMAD there. So in that cell line, but we haven't shown any anti-proliferation data in that cell line yet.
Yeah. I think the reason why we don't see like very pronounced effect on cell killing because we also compete the higher cellular concentration of TP. That probably means that we need to get sub-nanomolar potency or
the phosphorus goes all the way down, so it implies that actually having a serious effect.
One of the negative states of the kinase, so you won't see like big right shift, right? That's probably an indication.
Yeah, I think
like for another project we didn't see much, and then we got some nanomolar. We saw like a lot of.
In the other project, were you seeing the effects of inhibition on the downstream biomarkers though, like we are? Right, because we're seeing the phospho-SMAD being impacted, we're seeing the ID1, which is what you would predict based on, you know, it's not subtle.
Yeah, probably we can.
One mic.
Get some more. Do our best.
Yeah.
One of the things we can also forget, I mean, I don't know if a lot of this data is available, but I mean, the phenotype from knocking out the target versus the pharmacologically inhibited. So, I mean, I don't know if those need to be defined, because, I mean, maybe pharmacological inhibition is indeed cytostatic, and actually knocking out the whole target might be off of target. So, a lot of those still need to be defined. I don't know if there's a kinase dead version of that that people have thought about to really give a better readout for the phenotype of actually inhibiting the kinase function. But I think those are kind of little things to tease out. I mean, but we have good tools to really figure that out in vivo. I think.
Yeah, but still at the early stages of generating this kind of data, though, right? I mean, we're just starting to scratch the surface of these cell lines, of the different cell lines that are available. You know, so I think, you know, we're moving in the right direction, but there are still, you know, a lot of questions. And you know, I mean, obviously, it would be nice to see some cell killing here, but you know, we don't yet. So, yeah. Okay, maybe we should move on. To Alex, I think yours is this one.
Yeah, so we're able to present them from our side. We share screen.
Yeah, I think so. Let me see if I can pass the control to you. Oh no, that's actually you're going to be able to control my screen. Okay. See, yeah, Alex, I'm not sure where that is. Hold on.
Are you switched yet?
Stop share. If I stop share. No. Sorry, I'm just not sure where this is.
Okay, if it's too difficult, we can stick with you.
All right, this might be easier for this time.
Okay,
so,
let's do that.
All right.
Okay, so I think I'll start off by thanking the brain tumor charity. Supporting some of our work here with Zhongfu. Zhongfu is unfortunately, well, he's on holiday in Singapore. So we've put together some of his data, and I'm sure he can probably do a better job next time he is present. He's continued with some of the nanobret data, the latest batch of compounds. We had some really potent compounds here. This is just a summary slide of, say, half a dozen of them, with one on the next slide. Where we had some compounds down in the 13 nanomolar, sort of low sort of digit and double-digit nanomolar numbers.
Yeah. What's that? Yeah, those are repeats. Yeah.
Yeah. So the top and bottom are the 2 repeats.
Yeah.
He'll normally do 3 repeats, but probably waiting till he gets back. Okay, next slide. So the compound on the left, I think, was being prepped as a possible tracer for the nanobret. We're still trying to get tracers that we can do ALK2 and ALK5, well, all the ALK receptors in the nanobret system. But here we've also got some of the Charles River compounds on the right, 3015, 16, 17. Some of the alternative chemistry there, obviously not as potent as some of the other Charles River compounds.
Right, I think Sue probably will comment. Those were intermediates on the way to some of the.
Okay.
The two compounds on the right were intermediates, so we were removing that benzyl group and replacing another group on there. But.
Next slide. Actually, go forward again. Come back to that. We also did just today, actually, a thermal shift assay. So we were interested in an orthogonal assay before maybe we get the IC50s from reaction biology, where we could look at the relative potencies of the M4K series on ALK2. That would be the first column. So red would be the most potent, blue would be the least potent. We've cropped the data here, so we're not showing compounds of lower potency, of which there are some. And then we've compared that alongside with ALK5, ALK3, and ALK6. So for this assay, we make the purified proteins and then pop them in a fluorescence-based thermal shift 384 or 96 well plate assay. We've also done the type 2 receptor, ACTAR2A or ACVR2A. We didn't have time to actually process that set of data before presenting. We have that data recorded anyway. So I just highlighted with an arrow here 2 relative compounds, just to kind of give you an idea of what we're looking at. If we start with the M4K 3007 compound, you see that we have quite good potency on ALK2 with a 13.4 degree thermal shift. But when you look at ALK5, we have a number of 6.3 degrees and a blue color. And when you scroll across the other columns, the numbers relative are also blue, i.e. they're lower scoring potencies. I should say the scale for each column is independent. So a 17 degrees for ALK3 can't be the same as a 17 degrees for ALK2. They're independent scales. But what we're really looking for in the M4K series is compounds that will be red for ALK2 and, if you like, blue for any other kinase. So if we take the M4K 2009, which is the other arrow, it's also relatively red for the ALK2 and then blue for the others. So that's one of the most promising ones. I would say the M4K 3007 is still marginally better. These tend to correlate with a log scale of IC50s, so a relatively small drop-off on the thermal shift can actually correspond to a fairly big change in IC50. So there's no need to scrutinize these data too much. We can wait for the DiscoverX or reaction biology, kinome profiling, and IC50s. But if we just want to visually look through a set of data in a qualitative way across a large set of compounds, we can generate this data quite quickly with more or less any kinase that we can prepare protein for, including off-target kinases like ABL or RIPK2, things like that. Okay, we can share more of the whole table with you and put that or upload that to our server. Okay, the next slide.
You want to go backwards or forwards?
Forwards. We'll go forwards. Okay, so one of the questions Al had raised was, and maybe Paul is touching on this as well, what would be the ultimate or the final therapeutic? You know, if we were aiming for a combination treatment, do we need to target also the epigenetic change, or is that an exploitable genetic change, the histone mutation? And so we were interested in the EZH2 inhibitors, the methyltransferase, and also the demethylase. So John Fu was just doing some pilot or preliminary experiments. He's not here, and we haven't gone through it together, so I've just pulled out 1 or 2 figures without really knowing all the controls, the backgrounds, the replicates, etc. methods. So the point of this slide was really one thing that he found was that the EZH2 inhibitors alone didn't seem to have a big impact in terms of cell viability. And you can see that in the top row of this figure with the DMSO on the left and the EPZ6438, which is an EZH2 inhibitor on the top right. The M4K 2009 compound did seem to be quite effective. There's more data on the next slide, if you go to that.
Which cell line is this?
Sorry, I'd shuffled all the slides, so that's why I was going to present it from here. But this is the DIPG7 cell line, which Angel in Barcelona shared with us.
Right.
So this is more or less the same, similar data to the previous slide, but with dose responses. And he combined M4K 2009, one of the better compounds, with the different EZH2 inhibitors, which would be the middle 2, EPZ, and then the GSK 343. And then there's the demethylase inhibitor, the GSK J4. The M4K 2009 on its own has an IC50 in a cell line of about 1.2 micromolar, which I think agrees reasonably with what Jerome has in his other cell lines. The EZH2, it's not 100% clear if there's synergy or not. With one of the compounds, you can see here at the top, the IC50 was halved to 0.6 micromolar. There's still a margin of error there. And of course, it's somewhat dependent on the curve fitting. And if you have a data point on the top or the bottom of the curve that kind of skews the fitting. But there's potential there.
Alex,
yeah?
You know you have to treat with these the half-life of the methyl groups is a long time. So you have to treat like for a week or something, Cheryl does, before you see an effect. Did you know if Jung Fu did that?
Yeah, I know Cheryl told me it was 3 days for the methyltransferase and 6 days for the demethylases.
Yeah. And did he do that?
John Fu's not here for me to ask him.
Okay.
The previous slide with just the ALK2 inhibitor was, it says at the bottom 48 hours, which may not be long enough.
Exactly.
I know my student Liz has also done experiments just testing one concentration, and I know she did it for longer. And I think also we didn't see a great deal with the EZH2 on its own, but she's not done the combination. So I think consider these work in progress. And the bottom panel is PI staining, so this is for cell death rather than just cell viability or ATP content. And there we also saw both these ZH2 inhibitors halved the IC50. So there is potential that something could be going on. If you jump back 2 slides.
Does anybody have any experience with epigenetic combinations? This doesn't seem like having it is really much at all. If it is even significant, would you want to see like a big change?
Yeah. Do you mean combining 2 epigenetic inhibitors or a kinase plus an epigenetic inhibitor, Paul?
I don't know. I don't really have anything to benchmark it against, but even like the synergy, it should be more than just having. It should be tenfold increase. I don't know. I just try to think of any examples that we could benchmark this against. And I can't think of any. There must be some HDAC plus something, or bromidine plus kinase data.
Yeah, but they just tend to be cytotoxic to everything, so then you're not seeing your combination.
Yeah, that's true.
Right. I think Al might be onto something here in terms of, you know, the length of time that you treat with the EZH2 inhibitor might be important. So, you know, we might see more dramatic effects with the longer treatment. So.
The combination experiment can be very tricky.
Yeah.
I mean, you really got to do the isopolar. You got to do the dimension. You may pick a concentration that may not be actually that great. But if you do the whole grid of concentration.
That's what I was going to suggest.
Yeah.
We could talk to Richard. We could do it on the HP.
We could spray it.
Yeah, and he's got that matrix all worked out. He's done it with some other collaborators. So I think, and for us, we've done it on some of our projects. I think it would make sense to discuss with Richard.
Yeah, but I think the complication here is the difference in time, right? So two compounds will have difference in the activity time, right? One will take longer.
And we've done that with another project, though, right? Where we treated for like 5-6 days in the presence of one inhibitor, and then you add the second one.
Yeah, okay, yeah, that's what you have to do.
You know, sometimes that can make a big difference. So.
And going back to Al's point about the inactive controls, I mean, this is a great case for that because you're seeing a very small effect. So this is the inactive really useful.
Big brain pull there. I agree. Yeah, good point.
Well, I just repeated what Al said. So.
Yeah. Two big brains. We also, I mean, John Fu has a big set of slides on this, and I'll email him to ask what he's, you know, a little bit of background. But he also did some fact sorting with an exin staining. I think in this particular DIPG cell line, we saw 20% live cells and 80% dead cells. With a mixture of apoptosis and necrosis. So at least in this cell line, we had seen quite a lot of completely dead cells rather than just kind of cytostatic cells. I know Jerome, Jerome, you've just got this cell line now as well, so maybe you can compare them.
Unfortunately, I don't have it in my hands. Angel never got back to me, actually, so maybe I need to reach out again. And ask for it.
Okay.
But yeah, I would love to get it because it's been a grasp from even though I mean people I talk to at meetings say the cell line is peculiar but it can go in vivo. So.
Okay, so this slide was just looking at the M4K 2009 on its own. And so you can see here the range of replicates. So there was fairly good agreement between the repeats. And we got similar results with the cell titer glow and the PI staining, although you can see the curves don't actually reach 0 in the bottom row. Okay, sorry, I've messed up the slide order, Jeff. Do you want to hop forward 3? On this one, yeah, I think this is our last slide. So over the summer, the synchrotron's been shut, and we also had some virus problems for protein expression. The synchrotron opened, and we've had our first trip yesterday, and the viruses have already been regenerated, so we can be back to work now. The group here have set up about 58 crystal plates over the past month, with the half a dozen compounds shown here. Unfortunately, the yield of the crystals from this has been fairly lousy. There are a few microcrystals, particularly for 2009, and we can maybe use those for seeding and then grow some bigger crystals. So I'm sorry, on the crystallography front, we're disappointing for the September presentation. So I'll put pressure on the group to try and deliver in October. They're laughing at me. Okay, let's. That's the final slide from Oxford.
Great.
Of course, do send us updates if there are new exciting compounds and you want to crystallize those. Please just let us know the latest. And also, we find it's quite likely that the part of the molecule that is really solvent exposed could have the impact on the crystal packing. So if there's a group maybe in the back pocket that we're really keen to see how it binds or how it hydrogen bonds with the alpha C helix, then you know, even if there are some compounds with different solvent groups, you know, it could be that one of them will crystallize and one of them won't. So for example, I don't know if we have a derivative of 3007 with a different solvent group, you know, there's every chance, you know, maybe that would give crystals differently to this compound, in which case we could try multiple shots at finding the binding modes.
Unfortunately, we didn't have any, because this was one of the literature compounds, of course, so we were just making the selected number of compounds, and the synthesis of those cores was actually quite challenging, so we didn't have enough to do very much with it. So
yeah, I wasn't necessarily thinking purely of that one, but just in general, we can look at that from our side and try and maximize our success rates. Okay.
Okay. Thanks, Alex. All right. Let's see. This one. You all set, Sue?
Yep, I'm ready.
Okay.
Hi. So, yes, just a general update as usual. We've completed the synthesis of 8 compounds this period, which we're quite pleased about. And we've got several further compounds that are really coming along quite nicely. So we've started to make look at some design hybrids around sort of trying to overlay the pyridine series compounds with the blueprint compounds to see how we can incorporate some of the structural features of the blueprint type compounds into our main series. So we're working on some compounds around that area. So obviously, as Jeff has already mentioned, the PK studies, we've produced a quote, and we're just trying to sort out some timings on that. Just to let you know that this week, actually officially it's on the 13th of September, as part of our corporate social responsibility, we have what's called a Global Day of Service, where all members of staff are invited to participate in a charitable activity. And this year, as part of early discovery, that's the sites in Cambridge, Harlow and Hatfield, are dedicating this to the M4K project. So this has allowed staff to participate for up to a day, but across the whole week, because obviously being chemistry doesn't really allocate itself nicely to a day. So we've opened it up to chemists and non-chemists across all sites, and we've had around 70 to 75 odd people who've signed up for this to participate in design or synthesis of new targets. So we're quite pleased about that, and hopefully we'll get some output from it. So the aim obviously is to deliver more compounds, but also get greater engagement in the project, and also there's a staff development-ish aspect of this, where we're giving non-chemists, in fact some non-scientific people, the opportunity to go into our chemistry labs and see what we do as chemists. So it's been quite a busy week getting it all organised, but we're all. And the other aspect, I've got Sylvia sitting right alongside me, and I completely forgot to mention that, you know, she's actually going to help with the design side of things by running some CAD sessions, and Alicia at Chester Park as well is going to be doing the same thing. So yeah, it's really exciting. Hopefully we'll get some useful out of it. Oh.
From the whole team here at M4K, thanks very much for organizing this and the commitment you guys have brought to this program. I mean, to get 75 people to sign up and join, and we know that people will continue probably to work on this project going forward. I mean, this is fantastic. This is just this is more than we'd hoped that this kind of open science project would bring out to people. So thanks to your whole team over there. We've been very lucky with everyone joining in and all our partners coming forward, but this is real leadership from your team. So please pass on our appreciation to everyone.
Will do. Thank you very much. So do you want to move on to the next slide? So the next few slides, I'm just talking about just where we're at with some of the compounds that we've been making. So as you know, we've been focusing on analogues that are not the trimethoxy, not close to the parent compounds. We've been trying to find areas like heterocyclic replacements for the trimethoxyphenol rather than the more sort of subtle close-in modifications. There's a couple of new compounds here on this slide that we've completed the synthesis of, and they will go in the next shipment. So the M4K 3020 is the first of the heteroaromatic amides that we've prepared, and there's several further analogues around that area that are in progress. Move on to the next slide, please. So these are some examples of some non-aromatic left-hand side. So this was really stimulated by looking at quite a few of the literature compounds that we were considering at the start of the project. A lot of them don't have an aromatic ring in that area, in the sort of lysine binding area. So we thought it would be interesting to remove that aromatic ring. And on the right-hand side, you'll see an overlay. I think we've probably presented this last time, an overlay of compound. It's not exactly M4K 3023, but it's an analogue of that, with the blueprint compound showing where you see an interaction of the carbonyl with the asparagine, an interaction at the hinge with the histidine. Yeah, that's a lot. Yeah. Thank you. So there's a number of these piperazine type analogues being prepared with different functionality on the terminal nitrogen, because that's relatively straightforward to modify, obviously, so we can make a set of compounds. The next slide, please. So there's a few examples here. So the compounds that Alex was just talking about that were not particularly potent were the intermediates towards compounds like M4K 3019, where we've now taken that benzyl group and trimethoxyphenol on there. So these compounds are looking at modifying the core. So either having the pyrimidinone to replace the pyridine or the substituted pyridine, putting the carbonyl in the same region as either the methyl or the cyano, and that's that one, yeah. And then the two pyridone analogues, which give you a donor-acceptor pair at the hinge, which are related to some of the other compounds that we've looked at previously. So we have one further compound in that area, which is in progress. So that's the compound shown in the yellow box there. Next slide, please. So we talked previously about substitution. So all of the sort of the lead series have a substitution in the 3 position of the pyridine. So these analogues look to have substitution at the 4 position, but having a linker to put the aryl or non-aryl or heteroaromatic or heterocyclic ring into that same region. And you can see on the right hand side some of the examples of the compounds where you can get some interactions with the lysine through water mediated interactions with those compounds. So the first of those, the synthesis has been completed. There's the compound in the middle, M4K 3025, and we've got making good progress towards other analogues within these types of compounds.
Great.
Next slide, please. So some more new ideas we've been having, and this was really, so I noticed that the ICR paper has just been published. So obviously we'd seen this, the ICR quinazolinone paper previously, but it's now been published. And we were looking to see, so that was compounds at the bottom left. And we were looking to see whether we could use the quinazolinone core and overlay it with the blueprint compound above it and our own type, and then look to see how we might be able to attach a group to pick up the same sort of interactions you see on the right-hand side a couple of examples of where we've put a substitution. They're actually both the same group, but because of the way the orientation, the acyl pyrrolidine sits in a very similar position. So there's a good opportunity here to potentially generate a second series based around compounds of that type.
Great.
Okay. So something I raised a few months ago that Sylvia had been looking at was to generate some compounds by docking and using fragment-based docking. So she's going to go through the process that she's been carrying out and where we're at with that.
Yeah, thank you, Sue. So what I've been working is, let's call it lysine-targeted scaffold hopping. And if we move to the next slide, it's just a general background on what I've been doing. So it all started by trying to explore the area around the trimethoxyphenyl that we have in M4K 1062 or LDN 214117. Especially there are some sort of potential direct interaction with glutamate, the backbone of this leucine 281, even the gatekeeper, so this threonine 283, and also water-mediated hydrogen bonds with the rest of the pocket. So ideally, we want to replace this moiety, so this trimethoxyphenyl, and to gain additional interaction with the area that I put into this blue box. So if we move to the next slide, is part of the scaffold hopping protocol that I've been using. So I put together this library of that has been published previously of 502 fragments, and effectively I plugged it onto the moiety that is shown here. So in practice, the right-hand side of our compound. I restricted the library enumeration in terms of having max 2 hydrogen bond donor per structure, because we still have to get brain penetration. And I got roughly 1,329 hits. Then I started the refinement, and together with Sue, we decided to start with eliminating everything that had a log P that was too hydrophilic or too lipophilic, so just narrowed down the compounds with a log P within 1 and 3, bringing down the number of hits to around 609, and then I calculate the CNS MPO score. In this case, as you can see on the right-hand side of the slide, we have a summary of all the 1,300 hits that I've just described. And on the x-axis, we have the log P, whereas on the y-axis, the CNS MPO score from Pfizer. So we were in a sort of a lucky situation because a lot of hits actually scored more than 4. So we were decided to be quite strict and decided to pick compounds with a NPO score higher than 4.5. Therefore, we gathered roughly 450 hits. At this point, we still needed to sort of refine this number to decrease the number of compounds to actually dock. And if we move to the next slide, I have the second part of the protocol. So first of all, at the time, I removed the compounds that were sort of already prepared. And in that case, there was only one compound that came out as already under synthesis or being prepared. And what is further down is removing compounds that have structural alerts for toxicity. Those are alerts that have been published recently from Eli Lilly and BMS as well, narrowing down the number to 438 each. Now, finally, this was a okay number, but since we always have to be sort of time effective, I decided that it was probably easier to cluster these 400 hits to sort of pick the most representative compounds per each cluster and see which were the results. So eventually, I ended up with 96 hits. Each of them was represented in one cluster, and I docked them into ALK2 So in the next slide, I have some examples. So in this case, we have this left-hand side moiety that is a mix of hydrogen bond donor and acceptors. It has a very good overlay with our M4K compound 1062 that is shown in orange. Whereas we have a direct hydrogen bond with the lysine 235, and we also have a water-mediated hydrogen bond with the glutamate 248 via the NH of these new moiety. Exactly there. So these could be an interesting sort of development of the series we were working at the time, so the pyridine series. Other examples that we have in the next slide, I've just summarized some of them, of course, the most interesting. We have derivatives that sort of push with a methyl into the pocket where we have a leucine 261, so I'm talking with the left-hand side exactly, and they sort of bridge with the backbone of lysine 340 via water-mediated hydrogen bonds. Again, something new. If you look at the overlay with the trimethoxyphenyl, it's like we are closing the trimethoxy bit by these nitrogen-containing heterocycles. The rest of the molecule is again very well overlaid with the original trimethoxyphenyl-containing moiety. Whereas on the right-hand side, we have a slightly, kind of new interaction in a sense that the hydroxyl group is effectively hydrogen bonded to the gatekeeper, so this threonine, which is something that we haven't seen so far or we haven't investigated so far. So could be a possibility of further investigation. So we know that if we come up from that very vector or with an OH or hydrogen bond donor in that position, we can potentially gain an interaction with the gatekeeper. And finally, my final slide is just to show you that most of these fragments probably were not commercially available, like this one that I'm showing here, but they can sort of be a useful starting point to design targets that have a moiety that is currently commercially available, such as the one that we have on the right-hand side. And this one is currently in the pipeline and will be synthesized during this week, I suppose, because we have quite a number of people who joined. So. Yeah.
Okay, that's fantastic. It's really interesting stuff. So Sue, the intention is to make several of these?
Yeah, so the plan will be now to actually really look into, we did some very quick preliminary searching to see what was commercially available. And as Sylvia says, unfortunately, in the right sort of format, like with a bromine or boric acid or something like that, they're not commercially available. But then we need to look at how easy something like these or something that will give the same interactions. It's sort of inspiring more ideas as well. So yeah, we're planning on trying to pull some of these into the synthesis program.
That's great. No, I really like the approach. I think it's generating some interesting ideas for replacing the trimethoxyphenyl. So I think ultimately that will be nice to get some molecules that do replace that. So it's good.
I have a question. So Sue, I'm presuming that chemistry resource is going to be limiting for here and there. I just wanted to flag something I just emailed to Jeff, that Matt Todd, who was on faculty at the University of Sydney, has now been appointed as of last week Professor of MedChem at UCL. And so in that position, he will have resources. He was the mover behind open source malaria and getting a lot of distributed nets of students around the world to make compounds for malaria. But we know he's very keen on open. If we have low priority molecules that would be nice to have but not on critical path, I would like your guys' feedback, not now, but think about it, on whether we engage Matt he has a distributed community of chemists around the world, students. So for making weird ideas from virtual screening or from computational chemistry, it might be it'll be more difficult to manage. But on the other hand, it could be like 100 chemistry graduate students making molecules for it. And nothing more than grad students like is to test the hypothesis and have somebody do some assays for them that they can't do in-house. So I'm putting that on the table. He's just moved to London last week. He's very keen on the concept. It may be more of a distraction. That's for you guys to decide. But if you guys think that it could help, just let me know and we can have a meeting, let's say, in the UK between CRL and UCL as a possibility, okay?
Yeah.
I think, as you say, some of these areas where they're perhaps, a little more esoteric compounds, perhaps.
Yeah.
That could be the perfect opportunity to use something like that. Whereas, they might not be that trivial to make, but it would challenge some students.
There you go. That's exactly what I was thinking.
Yeah. That's a good idea. We'll have a think about it and what will be involved in doing it. Thank you.
Okay. Thanks, Sue. That was great. Wrong computer. All right, David. On the new template? No.
That's right. Okay, so again, just to start off with some 2009 data, some numbers that you haven't seen before. We've been looking at completing the data set for this compound, given that we're kind of designating this as our primary tool compound. So we finally screened this against the whole panel of ALKs. So we're looking at 1 through 6. And there's also the ALK2 mutants, the 3 that we're getting screened at RBC. We still don't have numbers for those. Those should be forthcoming in the next couple of weeks. But 1 through 6 is done. So something that we haven't seen before is we now know that ALK1 essentially equipotent to ALK2 for these compounds. ALK3, a little bit of a window. ALK4 and ALK5, a substantial window versus ALK2, and ALK6 somewhere in between. So I would imagine that for this kind of broad class of compounds, we're going to see that reflected in all of these structures. We're anticipating that for the ALK2 mutants, we should also see data which is similar to 1062. Given that if we're potent on ALK2, we should see this profile against 3 mutants, the R258G being the most sensitive one, as has been the case in the last 2 examples. We're also getting 2009, we're getting the IC50s for the handful of kinases we were hitting, which we found out from the kinase full panel, so that those are numbers that should also be forthcoming in the next couple of weeks. As far as the modifications to the trimethoxyphenyl go, we've been doing a number of very substantially divergent things over the last number of months. And we've been trying some very kind of out there modifications. We've been trying to stay close to the trimethoxy. And ultimately, nothing has seemed to be as good as the trimethoxy itself, with the exception of some of these very minor modifications, that is being a deletion of one of the O-methoxys in the central position, substitution with the fluorine. These kind of changes seem to be tolerated, and we're seeing actually some good biochemical screening numbers, and the numbers against the cell-based numbers, especially in the case of something like this fluorine substitution, aren't terrible. So we're just completing the series by doing the obvious methyl and chlorine modifications, seeing where they land, and maybe one of these will be a suitable replacement. But I think this will pretty much be the capper on this kind of work, and then we might look at exploring something else around some of the other motifs we've been looking at. So one of the other things that we have been looking at were the amide substitutions. Initially, we hit on this with this fluoroamide, which showed good biochemical potency. But in this case, for example, the cell-based data was not quite as good. What was interesting, however, is that if we take the fluorine and we put back one of the O-methoxys, as in the parent, we do have really nice potency in the biochemical assay, but we're also seeing good numbers in the nanobretnet too. So this is an interesting motif which we're going to take forward. One of the caveats here so far is we don't yet know, but if you look at the initial case of the fluoroamide, addition of the 4-position methyl dramatically decreased the potency on ALK2, and we're hoping that's not the case in this instance, but we're still waiting for the numbers to come back. Fingers crossed that this will not be detrimental. We've been looking at other modifications. Again, we've been doing a substantial number of variations around this, but largely keeping with the piperazine. or piperidine motifs. So here are just a couple of recent examples. We found that in fact some bulk is tolerated at this position. The piperidines do behave quite well, especially in the cell-based assay. And a number of variations like these bridged analogs, again, quite potent in biochemical, but not so much in the cell-based. So really the cell-based numbers are the drivers here because, you know, the biochemical does tolerate quite a few differences in these various positions here, but we ultimately want to look at that number. Around the piperidines, we've been doing a number of different things, trying to look at some kind of classic substitutions. In this benzoylic position, for example, we've run this series of small fluorine all the way up to this somewhat larger O-methoxy. And interestingly enough, when we have the small fluorine, we have good biochemical, and we have a pretty good number on the cell-based, and we have really nice selectivity. And this selectivity over ALK5 actually erodes as we increase the bulk at this position. And so we're down to a kind of very modest 40-fold here. And we also lose a little bit of the potency, but the cell-based stays quite good. So again, depending on how we make adjustments in the rest of the molecule, pieces like the fluorine or even O-methoxy might be useful down the line. Interesting compound here is also the one where we have the saturation, or unsaturation rather. And this is one of the compounds we're going to be taking forward into PK to take a look at how it behaves, and I'll touch on that a little later. We've also been looking at the oxopipridines over time in fits and starts, and what we're finding is actually this 2112 is a nice compound. Again, very nice cell-based activity, and very good in the biochemical, and a very nice window versus ALK5. So this is another compound. We haven't done too much in the series, but this is another compound we'll take a look at in PK. These are the tetrahydroisoquinolines, which I touched on last time. These compounds show very nice potency in the biochemical, especially in the case of 2089, and they also show very nice potency in the cell-based. These are in fact some of the most potent compounds we've made to date. You're looking at anywhere from 2 to 3, maybe even 4-fold more potent than 2009. The unfortunate problem here is that we are seeing some issues with the potency once we do substitute in the 4-position methyl. But that's not to say that this is still, I mean, this is a useful piece. We can work around that and see if we can recapture some of the selectivity and some of the potency in compounds where this 4-position methyl group is absent. So this remains to be explored a little bit more.
4-methyl really brings in selectivity.
Right, the selectivity. Right. So again, this is one of these things that's confirming this 4-methyl really does seem to be the linchpin for the selectivity. We see that over and over in all the series. It's just a question of does it ruin your biochemical and your cell-based activity. But definitely the selectivity seems to be uniformly conferred by that group.
So does this, if you dock it, does it dock differently?
In the docking, there's nothing is substantially different. Maybe the rings are a little bit twisted relative to one another, but there's nothing obvious as to why that would be happening.
But I think getting some crystal structure.
That's what we're hoping for eventually. Yeah, that's the ideal comparator for us.
I mean, there's one crystal structure with that methyl group, but with a different solvent exposed group. It was more the crizotinib type.
Yes, that's right. Yeah, and I've actually overlaid that one, and it overlays with 2009 quite well. There's virtually no difference again to look at. Right? So it's a little bit of a mystery, frankly, as to why this confers the quite substantial effect that it does at times. Here's a couple modifications again with the tetrahydroisoquinolines. Aromatization is detrimental, and here it is mixed and matched with the amide, and you'll see we have again quite good potency in cell-based and biochemical, but we have virtually no selectivity over ALK5. What's interesting is when we pair this with the nitrile, we do retain the potency, and we start to open the selectivity up again. So maybe the answer to that problem may be that the nitrile is the piece that we want rather than the methyl. So again, some possibilities exist around this motif.
Do you have any heteroatom in that position? Like, did you ever have nitrogen?
At this position? Yeah. Yes, we've done nitrogen. It's been slightly detrimental, or at least comparable. Not I think it was slightly detrimental, actually. Yeah.
And it was exemplified in the...
Okay. Limited modifications on the central aryl ring, which I covered last time. The only thing that is new is this compound awaiting screening, which we talked about. I think Sue mentioned marching the nitrogen around to this position to see if we can pick up anything at the hinge, if indeed it does orient that way. So we'll see where those numbers land once we get that data. And then this is the series that we've been kind of working on over time, trying to complete the 4-position substitutions. going from electron donating ultimately to quite electron withdrawing. We've completed the CF3. We see a drop-off actually in the ALK2 activity and a drop-off in the cell-based concurrently. And we have the fluoro compound, which is still awaiting data, so we'll see where this guy lands. But it seems for now that the nitrile is somewhat of the sweet spot because we see single-digit nanomolar in the biochemical, and we're also seeing a very potent compound in the cell-based. This is about twice or two to three times as potent as 2009. So we're going to take a look at this compound in PK, and potentially this might be a next-gen compound or something to look at seriously to move forward. So again, here's my little summary of criteria which we're trying to meet for a CNS penetrant drug. Nothing has essentially changed since I showed this last time, except we do have a new check mark here beside the hERG column here. So essentially, we're looking for a 34 margin between our hERG IC50 and the effective unbound plasma concentration. And now, thanks to CRL, we do have some hERG numbers for 2009. And it turns out that this compound is in fact around 8.25 micromolar against hERG. And if we take a look at the mouse PK numbers that we have for our Cmax was around 6 micromolar. And if we look at the mouse PPV and the HPPV and look at the free fractions, and we then calculate the unbound, we ratio it against the hERG IC50, we have a window currently in the mouse or human around 15 to 20, I would say. So we're not quite at the 30 cutoff, or better, ideally. If we could, right, the greater the number, the better for us. But we do have some ideas about what may be the culprit in the structure, and maybe this basic nitrogen, which is a classic issue. And we do have some ideas how to address this. And we will, in fact, be sending a number of compounds, which we already have in our tool set, to take a look at if maybe even some of those compounds will already address that issue. And if not, we have ways we can work around that. So, in terms of, yeah?
You did the assembly limiter slide that you saw?
Here?
Yeah.
At this position? Yeah, that's been tried once. It failed. So it's in progress. That'll come eventually down the line. So here's a number of compounds, 5 of them that are kind of in our next set for PK profiling. We haven't really looked at the amides, and I think this is a nice candidate to do so given its cell-based potency. We haven't looked at the nitrile, so this will be the first compound that will go in. As I mentioned, the oxopipridines, again, nice and potent, and a couple of the examples where we're looking at the piperidines unsaturated here and methylated at this position. So we'll see what these look like, and I think this is our first set beyond the first one that we did. Right? So second set, right? Yeah. There's a couple other stragglers that we could look at for PK, seeing the influence of this O-methoxy at the benzylic position and these pairings of this tetrahydroisoquinoline with the amide and nitrile with the O-methoxy. So those are potentially a little bit down the line. To address the hERG issue, here's just some compounds that we're currently assembling the set, which we hope to send out today or tomorrow, which could address the liability, solving the problem with these classic methods of bulking up around the nitrogen, maybe different chemotypes, conversion to the piperidine, this kind of distal bulk to the nitrogen, etc. So these are some classic ways we can potentially address the hERG issue. We'll start with these compounds, again, see what kind of numbers they give, and then we can work from there. And finally, I think Alex touched on this, the tracer development for the ALK5 nanobret. I think Alex and David were looking at putting together some linkers in which they could attach these DIPG-based fluorescent probes. So this is ultimately what they're aiming for. And I just started to take some of the 1046 we had in-house because it does show good ALK5 activity in the luciferase cell-based. And I started to attach a couple of the potential linkers where after removal of the Bach group, this fluorophore will be attached. And so far, this one has yet to be screened, but 2144 has been screened. It does actually show good ALK2 nanobret activity. So this is hopeful that the ALK5 cell-based activity will also be quite good, and this might be a useful linker or some derivative thereof to make our tracers down the line, which would open up the ALK5 nanobret for us. And that's where it stands right now. So one of the other things I should mention is that a colleague of mine, Brian Wilson, has a student who's going to be working for him for a period of time. Over the coming months, and she'll be actually doing some of the chemistry around the southern portion of the molecule. So we're going to try and do some kind of expand the range of substitutions because we've been largely in the piperazine and piperidine area. So we're going to try and look at kind of diversifying what's in that solvent front, which may address not only the hERG issue, but may also open up some potential binding and whatnot. So we'll be working on that in the coming months.
And give Alex some options too.
That's right.
That's right.
Absolutely. So
phenotype or knockdown. Pharmacologically, you might want to think about post-hoc status if you just have genes of the pharmacology. So, in fact, let that group is tolerated. And if it's taking a post-hoc and then defining some of the things, instead of actually lentiviral infections. So I look like the cells are very delicate, the DIPG cells. So maybe this is a good way of actually not only looking at pharmacological inhibition, but also actually knocking out the target to better define.
Yeah, but that would be a completely separate full-fledged SDR.
Yeah.
But you, I mean, I'm not against, but I'm just saying, just keep that in mind.
Yeah.
You have to have somebody full-time on it for a while.
Yeah. The chemistry is quite involved to make those PROTACs. So, but yeah, it's an interesting idea. Certainly something we can.
But do we know that we can't do the knockdown in those cells?
No, we don't know. I mean, I presume we probably can do the knockdown. It's been done in other cell lines. So.
I don't know.
No, it's possible. I mean, I've CRISPR 10,000 of cell lines. There's some issues depending on the cell line. Sometimes easier than others, but it's definitely doable.
Great, thanks, David. Great update as always. All right. 11:27. All right, perfect. All right. So again, I just wanted to come back and remind everyone kind of where we were, because we do have new people joining the project. So I just wanted to remind everyone what the key issues were when we started the drug discovery effort here. The freedom to operate, there were 2 patents from Harvard that covered the lead molecule and several of the close analogs. We've been able to get around that already. We needed to improve the potency. We tested the lead molecule in our hands, and it repeatedly came out less potent than what was reported, and against ALK2, and we definitely wanted something that's a little bit more potent than that. Certainly the compounds have to cross the blood-brain barrier. We know that the lead molecule does, and so we were using tools like the multi-parameter optimization scores. We're hoping to get the MBCK MDR1 online. And of course, as I always say, time is of the essence. I think that, getting to this early proof of concept to prove the mechanism will work is something that's really high on all of our list of priorities. So, and I think we're moving in that direction. We've gone through the SAR, so I think this is just redundant at this point, but, we wanted to try to replace the trimethoxy, look at different hinge binding motifs, alter the solvent side binding group. But because the lead molecule had good properties, good PK, good brain penetration, we purposefully have stayed very close to this scaffold. So that's why all the molecules that you've seen from David are really, really close to this. We're just trying to improve on what was already known. And M4K 2009, where we moved the methyl from this 2 position in the lead molecule, and you see that it was not that potent against ALK2, to the 4 position. You see that we get a 5-fold improvement in the cell-based potency and maintain good selectivity. Not quite as good, but still probably adequate in terms of a proof of concept molecule. So we moved that compound through our cascade. We got good PK in mice, showing good clearance, certainly a similar overall profile to that of the lead molecule, with even a superior Cmax and AUC, although the bioavailability was higher than 100% in this case. So, I think that this leads into the fact that Charles River is going to repeat some of this work for us. But given this data, we moved on. We moved 2009 and several other compounds into brain exposure experiments. And indeed, we were able to get about 6 micromolar into the brain at 2 hours after a 10 mg per kg dose. So pretty good, with brain to plasma ratios of about 0.4 to 0.3. So not bad. 2045, a slightly less potent compound, is also, I think also has a pretty promising overall profile as well. And one interesting thing that we did discover is that efflux generally gives poor brain exposure. This molecule 2043 is shown here, and you see that it has about a 2.5-fold efflux ratio, and that molecule shows a very poor brain exposure. So, getting that KCO2 data is going to be important for many of our molecules. And then Charles River, we're able to look at the mouse plasma protein binding of 91% for this compound, and the brain tissue binding of 99% for this compound. And we're able to generate some, basically the fact that we should exceed the EC50 out to about 8 hours in plasma, but only about out to 2 hours in the brain, if the unbound fraction is in fact what's relevant. So I think that that's also an interesting piece of information. Given that, we decided to continue evaluating M4K 2009. We evaluated it across the full kinome. At RBC, we hit about 10 of 374 kinases at 1 micromolar, of which 3 of them were the ALK 1, 2, and 6. So, not that surprising. We kind of already knew that, but it's good validation for the assay. We also hit ABL, DDR1 quite hard, and TNIC quite hard. And as David mentioned, we're getting full IC50s for this list of kinases, and we're waiting for that. The compound has also been scheduled to be fully profiled across the kinome at DiscoverX, a slightly different platform. Yeah, we're waiting to see those results. We also evaluated M4K 2009 against a SERAP panel, one that we use here relatively frequently, which is the safety screen 44, and we also profiled against HERG. So David showed you the data that we generated that shows that we do bind to the HERG channel at about 8 1/3 micromolar. That's kind of on the cusp of being a risk. Obviously, if we wanted to move this compound forward into further preclinical studies, we have to do dog telemetry studies and other follow-up studies to be able to mitigate that risk. But at that level, it's definitely not a showstopper at this point. We also, in the safety screen 44, identified several targets that we do hit relatively hard. Certainly the cannabinoid CB1 receptor at 10 micromolar, we're inhibiting it at 97% inhibition. I don't think any of these are showstoppers for a proof of concept molecule. And we did discuss this a little bit in our meeting with the folks from Charles River, and I think everyone's of the same opinion that for a proof of concept molecule, this profile is okay, but to move it beyond that, we would need to do more tests and certainly get more opinions in terms of the relevance of these targets in a DIPG setting. So we're trying to mitigate this risk by profiling 10 other compounds, as David mentioned, against HERG, and we're getting quotes for full IC50 curves against these receptors, but so far it's looking like that's going to be quite expensive, and at this point it may not be worthwhile to get that data until we see whether or not we can truly generate a proof of concept data with that molecule. So the target product profile of M4K 2009 is highlighted on this slide. We're getting through, we're checking off many of the boxes with this compound. We're waiting on the Ames data. There was a hold up in shipping, otherwise, got held up in customs for a couple of weeks in China. But we should have that data for the next meeting. And, essentially, again, we haven't identified any showstoppers. So we are scaling up this molecule to try to be able to start looking at some in vivo proof of concept models, both in the FOP as well as in hopefully eventually some DIPG models once we finalize what that'll look like. We also, as David highlighted, identified some new compounds that we're going to push into in vivo pharmacokinetic experiments. These are generally have pretty good overall profiles. I think I've actually mixed up the MLM and HLM columns here. These numbers should be here for these compounds. And we need the KCO2 values for these compounds, but we're pushing them in at risk since they do represent the kind of key structural features that we've identified that, kind of bringing potency and or selectivity to our series. So it'll be good to get an initial in vivo read for these molecules to see how they look and whether or not they're going to be able to compete with or even displace M4K 2009. And then David did a really good job highlighting some of the more recent compounds that we've identified. These things sort of looking a little bit like the blueprint piece, where I think this looks quite interesting, as well as with the amide combining it with the tetrahydronaphthalene, we can see that we're getting really quite potent molecules. So incorporating the C4 methyl into these analogs is high on our list of priorities. The nitrile, as David mentioned, also looks extremely promising. And if we can find a way to improve the selectivity a little bit, or if these things actually do have good PK, I think we're looking quite good there. And then we just recently got this compound 2149 that allows us to look at a trimethoxy replacement in the presence of the C4 methyl, and if the potency stays the same as kind of these analogs, then I think that'll be another alternative that we'll be able to move into at least evaluating in vivo pharmacokinetics and starting to think about whether or not this can displace M4K 2009. But I think we're generating a list of really interesting compounds that are potent and selective, and I think we're moving compounds through that pipeline quite well. I do want to remind everyone that M4K 3007 also remains a very interesting compound. I think Alex showed as well that this is pretty selective across not only against ALK5 but against the other ALKs that they've looked at. And this does come from the blueprint patent essentially, and so we don't have freedom to operate, but we are going to evaluate this compound in vivo because it could represent also a very good tool compound to demonstrate this critical proof of concept that we need. So we do have quotes for running both of these compounds through the CRL in vivo protocol, which is going to look at both the plasma and the brain. And Owen's approved it, so we're going to move forward with those experiments, and we should be able to get data for M4K 2009 in another set of hands that will verify the data that we already have for that compound. And M4K 3007 is an interesting compound in its own right, and I think it'll be great to have that data as well. So the next steps, we want to continue profiling M4K 2009, scaling up to 10 grams. As I mentioned earlier, the quotes were quite costly, mostly because of the cost of a key starting material. It was more than half of the cost for making the compound. So we're exploring alternative routes here at OICR, and we'll see how that goes. I think we should be able to identify a better route than or a cheaper route. Let's call it.
What does expensive mean?
Well, more expensive than what we'd want to spend for making the 10 grams. So it was about 10,000 Canadian dollars to make just to buy the starting material to make 10 grams. I think we can cut that by a third at least by going to the dibromo analog that and Sue.
So it takes $3,000 but spend 2 months to figure out.
It won't be that long. It won't be that long. I mean, the timelines for the CRO that I got quotes for, it was a couple of months anyway before they could get to it.
And so we can't do a mouse experiment until.
Oh, we have some material already in hand, so we can. Yeah.
Okay.
Yeah, but again, it's not going to take very long. Probably by the next meeting, we'll have some of these answers.
But because synthetic road is going to be developing that synthetic road ongoing is going to save us money as well.
Yeah.
I mean, it's just this one time off.
Because if you come with a cheaper road now.
I get all of that. I'm just pushing back. We need that. I don't care about more self-line experiments, right?
Yeah. Yeah.
If we want to do it in the bone model that this pen lady has, we got to do that soon.
Oh yeah. No, I know.
So it's not stopping us.
It's not going to stop us. We'll have the material for that long before we need it for that experiment. We need to finalize the MTA. She needs to order the mice and quantize them, get them.
Does anyone have any mouse models for any FOP or this up and running?
Well, these guys do have it up and running, but it's, you know,
That's not up and running, that's in theory up and running.
No,
right?
no,
it's up and running. You
it's
just
up
gotta
and running.
order it. With all these vendors, you have to order the animals, get them in line, get in the queue. They're always doing this kind of stuff.
Yeah, it takes a little bit of time. There's a time lag between let's do it and let's.
Yeah, so our Let's Do It was 2 months ago, right?
No, it was just a couple of, like a week ago, I think we had the
But
first.
how long have I been back?
Yeah, yeah, so, you know, I think things are moving along there pretty well. You know, I agree with you, getting something in vivo sooner rather than later is good, because we do need to generate that proof of concept, but, you know, I think the doing it stepwise, getting the FOP data to make sure that we're on target and impact disease. I think we all agree that that's an interesting way to go while we figure out what the best DIPG model is and how to get that.
We'll never know what the best DIPG model is. I would argue if we get efficacy in that FOP thing to show that it gets, and so I would start, if we don't get a result on a cell line or this is going to stop us, I don't think so.
Yeah, I mean, I guess we could show efficacy in the FOP and safety, and we know we're on that target.
And then make sure in the animals that the compound got in the brain and reduced signaling.
Yeah, and Javad is working on that, and so.
And I'm not so much sure. You know, we should talk to the regulators then, because, you know, starting I don't know if I'm right, because I'm just speaking out of my you-know-what. But you have efficacy in them all, you know it hits the target, you know it got to the brain, you know it inhibits signaling, is a weird-ass DIPG model made from a cell line that was coaxed to grow, right? Is that going to stop us, or should it stop us? I don't think so. The genetic evidence for this mutation is so bloody strong in these patients, right?
Yeah,
so,
it's definitely.
I tell you, that's why I'm thinking, you know, as soon as you start to see the FOP model work,
we can at least talk to the regulators
right then.
It wouldn't hurt. Yeah. Yeah. I mean, I don't.
Because the model is so contrived, right? Yet the human genetics is so strong. It's not like you do an experiment where you think it's upregulated or downregulated and maybe it'll work. This is a bloody hell. This is a driver mutation.
Yeah.
So what did you decide on?
We're doing it internally, at least.
You're doing
exploring chemistry. So starting from the dibromo instead of the chloro.
We should have that answer soon. That's not a drawback or a hold up any measure. Yeah.
No, we've shipped compound. We have what 500 mg, something like that. The compound in hand that is doing for all of the tests, all of these preliminary tests, and by the time we're ready to ship with the FOP model, we'll have enough.
And in the near term, we can always make several hundred mgs of that compound. It's not that difficult. With some focus, we can, right, a couple days.
So the key is that efficacy model. How much do you need?
I don't know. We're still getting the MTA in place, and then we need another call to finalize the experimental design. She is able to look. So we talked about looking at 2 different doses, and then harvesting and getting the material to be able to look at the phosphorus SMAD. She also has some other markers that are dependent on ALK2 that they've identified in the model, and so, you know, she's willing to look at those as well. So, you know, I think we need the MTA in place, and then, you know, we'll finish designing the experiment. Yeah, that's coming. Yeah. So. Yeah. Okay. Yeah. So we're getting the IC50s for the off-target mutant kinases. Our in vivo meeting that we had with the folks from CRL, we discussed both the dose escalation and the MTD. So we all decided that we would do the dose escalation right away, look at how linear it is across that PK range, and then based on that information, we'll start to design the MTD. And we can either do that here, or perhaps we can farm it out, and we'll have to discuss that. But once we have this information, we'll be able to move forward with that. So that's been scheduled, and the compounds were shipped out. So we should have that for the next meeting. In vivo safety efficacy. So this FOP model from Eileen at UPenn she's got this knock-in R206H model. The MTA is in progress. So, you know, we're hoping to, again, establish efficacy in altitude-dependent model, and hopefully some PK/PD as well that'll help us inform any in vivo study in the DIPG model. So the DIPG 007 model from Chris and Engel, you know, these discussions are still in progress, and Jerome, you know, I know you're working hard to try to generate models as well.
Yep.
So, you know, and then, you know, there are some other DIPG models around. We'll have to reach out and start figuring out, you know, even if, you know, maybe we can move ahead without that data, I still think it would make a stronger case if we could at least show some efficacy in one of these models. If it doesn't work, I think, you know, if we could convince the regulators that the FOP model is more validated, there would still be a path forward, but it would be good to show some efficacy in a model. Javad and his team are evaluating M4K 2009 versus a panel of DIPG cell lines, and they're also looking at brain exposure using their microdialysis, so real-time brain exposure in the microdialysis model, and so that'll be interesting to get real-time brain information moving forward. And then Max, we had talked about a more thorough IP analysis for this compound, and I know it's something that's on your radar.
I actually searched the compound to see if it was exemplified in any patents anywhere in the world, and the answer is no. So it's clear from that perspective. As you know, though, there are genus-type claims that we need to look at, and we're pursuing a potential contribution from the IRAC program from the government to support that. That has gone nowhere, so we're going to talk about next steps, I think, after this meeting. But
okay.
Getting a more thorough search on that compound.
Yeah. And then, you know, I think David and Sue did a great job showing you we are generating more exciting analogs. We're testing them. I think, you know, we still need some of the ALK5 data. I know that there's been a problem. You know, the Oxford end to try to generate that data. They've got some issues with the assay. And we're working hard to try to get an ALK5 nanobret, but, you know, driving the SAR without that ALK5 data has been a little tougher. But we are getting at least the in vitro data from RBC that's helping us drive some of it. So the in vitro ADME is ongoing. The profiling here at OICR, and the PK and hERG evaluation that we've already talked about is ongoing. And then I think we saw some really good plans on all sides in terms of continuing the SAR in a couple of different series, identifying a better M4K 2009 analog from the lead series, identifying a backup series, whether or not that's 3007 or some of the hybrid analogs that Sue and the CRL team are generating. I think that still needs to be determined, but you know, we'll see in the next little while how all of that chemistry plays out. You know, again, I think, you know, the team's making great progress, and we're getting the right data for this compound. So.
Have you decided MTD design yet? Are you going to like single and multiple dosing in the MTD or?
Yeah, I think a lot depends on you know, the dose escalation results. You know, I think that there were some results that I saw from some of the compounds that perhaps had higher exposure. That some of this data was generated at ICR, like an N-methyl compound that was toxic, right? Mm-hmm. And so, you know, let's see what happens with this dose escalation, if it's tolerated, if and what, you know, what our linear range is, and then based on that information, I think we'll design the MTD. Ahmed has done this a couple of times. So. Yep. Okay, I think that's it for today, unless there's any other points for discussion, comments, questions.
Are you going to get for your dose escalation? Are you going to do any brain concentration testing during that?
We discussed that, and no, not at first. You know, we'll just get the PK first. It just gets expensive. Yeah. All right. Okay. All right, thanks everyone. Thanks for another great meeting, and we will. I guess the next meeting is October 4th, so hopefully I think we should have some good results for that. So thanks again.